And we are back. This is going to be chapter 14, part 2. We have explored the world of the mitochondria and have become familiar with its inner workings. So now let's go ahead and take a look at the chloroplast and its ability to utilize energy, both generating it and hydrolyzing it. All right, now it's very common to compare the mitochondria and the chloroplast. For many of us, when we hear the chloroplast, we think about photosynthesis, we think about plants, we think about basically about any type of organism that can do autotropic reactions, meaning that it's able to generate food from sun rays, sun energy. Now, what do chloroplasts and mitochondria have in common? Well, both of them are used to prove the endosymbiotic theory. Both of them were their own individual living entities billions of years ago. Both of them have their own DNA. Both of them will have a double membrane. And when we look at those membranes, we see that for both, the outer membrane is very permeable, has large openings so that molecules can pass through, while the inner membrane will be less permeable. And both of them will have a narrow intermembrane space between those two membranes. Hold on, let me get that right here. So both of them will have the outer and the inner membrane with very similar characteristics. Now, as you may recall from the mitochondria, it had a central part that had the little infoldings like this, and the inside part we would call the matrix. Well, the version of the chloroplast that's going to be the matrix is going to be called the stroma. And you can see right here in the illustrations, you have your little chloroplast floating around. They're the green little items right here. Here's a little illustrated example one. And you can see that they went ahead and they drew out the double membrane. And you can see the inner membrane space as well as the stroma section, which is the mid part of the organelle. And now what we see happening is that there is a very specific difference structurally between the chloroplast and the mitochondria. And that's going to be in the fact that chloroplast has an extra compartment. It has a stack of flattened disc-like structures that we call thylakoid. And these thylakoids, as I said, are going to look like little stacks. You can see it on the picture right over here. They are going to layer on top of each other, and they're going to be able to connect their inner workings with each other, and therefore they'll have this area called a thylakoid space. And the thylakoid is going to be extremely important because what we're going to see in the chloroplast is that the majority of the work for photosynthesis will not take place in the inner membrane like we saw the mitochondria do. Instead, the majority of the work that the chloroplast is going to be conducting, especially when it comes to photosynthesis, is going to be done within the thylakoid region. So it's a huge difference, and obviously the fact that the thylakoid is there and the fact that it'll be doing a lot of the energy-driven mechanisms and reactions that we rely on. Well, not we, but photosynthetic organisms rely on instead of being done by the inner membrane. All right, so what exactly is photosynthesis, right? So if we're going to go about and we're going to define it, it basically means that we capture light energy and then we utilize that to produce ATP and NADPH, so our activated carriers. And then it's not the final step because once the NADPH and ATP molecules are formed, we see that the process will continue on and it will be utilizing the energy in ATP and NADPH to convert carbon dioxide into organic molecules that will serve as precursors for sugars. And this whole process of converting CO2 into organic molecules is going to be called carbon fixation. So within photosynthesis, we see that we can categorize those organisms as autotrophs because they're able to generate their own food, whereas we, of course, are not able to do that. So we are going to be heterotrophs, meaning that we are consumers and we have to consume other plant or animal products and thereby take the energy from their source into ours. But we've discussed that matter before. So on your slide, I wrote down for you that photosynthesis occurs mostly in plants, algae, as well as bacteria that are able to photosynthesize, which we call cyanobacteria. And the photosynthesis is going to be very effective due to the fact that we have the chlorophyll stacks we have the thylakoid stacks within the chloroplast. And what the chloroplast is interested in doing is it's able to capture blue and red wavelengths of visible light So what we're going to do is we're going to have chlorophyll embedded within it. And chlorophyll is basically going to allow the absorbency of the energy from the blue and the red waves. In a little bit, I'll show you a photospectrum so we can kind of talk about it a little bit. But either way, chlorophyll will be the one that will be attracting the energy from the sunlight. The whole process of photosynthesis is divided into two main stages. You're going to have a light reaction, which, as the name indicates, will have to have exposure to the sunlight at a certain point. And the products of that will be ATP, NADPH, and oxygen will be produced in that particular stage. And then the second stage, it's light independent. We call it the carbon reaction. You might have also heard of it as the Calvin cycle. Either way, this is the final stage of photosynthesis, and this is then when we take the products from step 1, including our ATP, our NADPH, plug in some carbon dioxide, and we're able to make our precursors that will eventually lead to the production of sugar, aka food for the plant. Now, in case you were wondering, plants are able to both photosynthesize and do aerobic respiration, because as soon as they're able to make the sugars, those sugars will get plugged in right away into our aerobic respiration, and they'll undergo the glycolysis, the Krebs cycle, and the electron transport chain step. So both of these kind of feed off each other. All right, so here's just a little illustration showcasing our two stages of photosynthesis. The entire chemical reaction has been written for you right on top of here. And basically the input is going to require carbon dioxide, water, obviously light energy, and then the end products are going to include glucose, C6H12O6, and then a byproduct that's being produced is going to be oxygen. So based on the formula, you can see that plants are carbon dioxide consumers and oxygen producers, whereas humans are oxygen consumers and carbon dioxide producers. Remember how we generate carbon dioxide within the Krebs cycle of the electron transport chain? This is one of the main reasons why it's such a symbiotic relationship between plants and ourselves, because basically plants produce the oxygen that we require and we produce the carbon dioxide that they do. And they vice versa utilize the byproducts, the waste products that we don't care for. Over here, it just reminds you that stage 1 of photosynthesis is the light reaction. And for that, the location will be our thylakoids. And the main purpose is to utilize exposure to the sun's energy and convert it into ATP and NADPHs. Now, I don't have to tell you, because by now we should be little experts on this, that the ability to produce ATP and NADPHs is so because they are both activated carriers, and they're basically harvesting the energy from the sunlight in an intermediate form until it's ready to be passed on, which is what occurs in stage 2. That is going to be our light independent / carbon reaction. This will occur in the stroma of the chloroplast, and now we see that we can take that ATP and NADPH that was created in step 1, and we can utilize it to convert carbon dioxide into simple sugar. And of course, our main goal is to get to glucose. Here's a look at the wavelengths and what we like to call our electromagnetic spectrum. It's the visible light. And what we see happening is that within our thylakoid stacks, we have our chlorophyll. Chlorophyll is what's going to be able to absorb the energy from the sun, and it favors the red and the blue wavelengths. It's written for you right over here. Red and blue wavelengths. Blue is 430 nanometers, whereas red is 660. So that's the scale that we're looking at. The color that it can absorb extremely poorly is green. So what we end up doing is that when you look at plants, they tend to look green to us, not because green is the color that is being utilized by the chloroplast. It's because green is actually being reflected back. So it's not absorbed, and because it's reflected back, that is then the color that we associate most of the time with the leaves or the various different plants. Now, within chlorophyll, and you can see a little structure right over here, its power or its ability to be able to detect the energy and absorb the energy from the sun all comes from the fact that it has a little porphyrin ring. And this porphyrin ring is basically a central area that is surrounded by a cloud of lots and lots of electrons. And what's going to end up happening is when these electrons become exposed to the energy of the sun, they get really excited, and that excitement is then converted into a form of energy that we can utilize within photosynthesis, obviously starting with stage 1, which will be the light reaction. So here's just another look at our photosystems. Now, the way you want to think about it is that a photosystem is just going to be a large protein complex, okay? So right over here, we could start on the right-hand side. We can see one illustrated for us. And the way the photosystems are going to work is that you have a region that we're going to go ahead and we're going to call it an antenna complex, and we're going to have a reaction center. And collectively, these are just these large multi-protein complexes. And the way it's going to vary is that your antenna complex is right over here, and it's also written for you, have all the accessory pigments that can capture the light energy. So they're the ones that will interact with the sun directly. And once they're able to transfer the energy from the sun over to the chlorophyll molecule, we see that it's almost like playing a little hot potato, very similar to the electron transport chain. They're going to be moving the energy from one area, one antenna complex to the next, until it eventually reaches what we call the reaction center. Now, the reaction center is where you have a very special pairs of chlorophyll. We call it the chlorophyll special pair. Ha ha ha. There's specialties all in the name. Now, what's so special about this pair is that it turns out that it's actually at a lower energy level versus the antenna complexes. And because it's at a lower energy level, it makes it more favorable for your energy to stay there, not the electrons, the energy to stay there, the energy that came off the sunlight. And that energy will stay there. And then the next step is going to be, and this is the part where we're going to take a look at the picture below us. So go ahead and take a look. The special pair in the reaction center is interacting with the energy courtesy from the sunlight that's been bouncing down the antenna complexes. And what we see happening is that there's going to be a transfer from the energy into our electrons and our electron carriers. So once again, we see the introduction of our electrons, and we also see the introduction of our little mobile electron carrier. So this is where we're on our way to take a look at our NADH molecules as well as the ATP molecules. All right, now this concept, as you can see, it is a setup system with various stops. So it is very similar to our electron transport chain, and that is also why stage 1 of photosynthesis is often referred to as an example of oxidative phosphorylation. There's two main differences though. One is that the energy that comes to fuel the system or to start the system does not come from high energy electrons courtesy of food bonds. Instead, the energy is going to come from chlorophyll that was able to absorb the energy of the sun. And the other thing I want to mention to you is that as we continue to follow the steps, we're going to notice that the final acceptor will not be oxygen. Instead, it's going to be NAD positive, which means that it's able to harvest that electron and generate NADPH, which is, of course, our activated carrier. So please keep that in mind, is that our source from energy is not going to be an electron initially, because we are not getting it from pulling apart bonds from food molecules like we do in aerobic respiration. Instead, we're taking energy from the sun, courtesy of the chlorophyll molecule, and we're moving that energy step by step until it gets to a reaction center where it has a lower energy point, so it's more favorable to hold on to it. And from there, the energy is then converted over to an activated carrier. Thank you, NADPH. Now, just to be clear, and I probably should have mentioned this to you before, When you're looking at your first step, stage 1 with our light reaction, that is where the photosystems are going to come into play. So our whole discussion on our previous slide with the fact that we were looking at an antenna complex and a reaction center, this is all occurring within the first stage of photosynthesis, which is going to be our light reaction. And the photosystems, turns out they come in two variations. We have photosystem 2 and photosystem 1. And a funny story behind this, if we're looking at the order that it occurs, photosystem 2 occurs first, and then photosystem 1. That's why I wrote it like that on your PowerPoint. It's not because I don't know my Roman numbers, it's all because of the fact that photosystem 1 was the first one to be discovered. So they named it photosystem, and then when they discovered that there was a second one, but it happened earlier, they just went ahead and they just named it photosystem 2. So it's all on basis on which one was discovered first. Either way, what we're going to see is that the goal of photosystem 2 and 1 collectively is to go ahead and harvest the energy from the sunlight into our ATP and NADH molecules. And as we're doing that, we're also going to be able to generate some oxygen as a waste product along the way. So go ahead and take a look at your picture. This is figure 14-35 and 14-36. And as you can see, what we're going to happen here is that the first one to occur, as I mentioned to you before, is going to be photosystem 2. And as the light energy comes down and it's captured, we're going to go ahead and see that the high energy electron is going to be transferred over to our plastoquinine, which is an example of a mobile electron carrier. So here we go. So we had our light energy come down. We had it interact with our reaction center. We could able to convert it over to an electron, and that electron is going to move over to our plastoquinine, which, as I mentioned to you before, it's a mobile electron carrier. It's kind of like a stepping stone. It's very similar to the ubiquine that we saw in the mitochondria. From there, we're going to transfer the electron to a proton pump. As you can see right here, it's pumping the protons through. This should look very familiar to you because we just discussed our little oxidative phosphorylation in our ETS step. And as we're seeing, this proton pump is going to be called cytochrome B6F complex. And fun fact, it's very similar to the cytochrome C reductase complex that you would go ahead and you would find in your mitochondria. The last step for photosystem 2 is going to be the fact that we're going to harvest the energy of our proton pump, and we're going to utilize that for movement through the ATP synthase, high enzyme that we know so well. And as you can see, the movement of the of the protons is going to be fueling the phosphorylation of the ADP so that we can go ahead and generate ATP molecules. So that is our first activated carrier that we are after. We are not done yet though, because we still have photosystem 1 to go. And in photosystem 1, and this is the part where you take a look at the illustration that's more towards the right-hand side, we see that you have a high energy electron that's going to be passed once again to a mobile electron carrier. This one we're using though has iron sulfur in the center. It's ferrodoxin right over here. And the ferrodoxin one is going to be able to allow that energy transfer to proceed quickly, so that in this case we're not generating ATP, but we're going to link it with NADP so that we're able to go ahead and activate our NADPH. So there is our second activated energy carrier. So notice how at the end of my light reaction, I have been able to generate ATP molecules as well as NADPH molecules. Along the way, as we proceed through photosystem 2, we do see that one of the steps is going to involve the fact of generating oxygen, my apologies. It's generated due to the fact that there is what we call a water splitting complex that's associated with a reaction center that we find in photosystem 2. Now, this water splitting complex is basically an enzyme that has a manganese cluster towards its center. Here you go, it's pictured. And what we see happening is that the oxidation center is essential because it's going to allow for the electrons to kind of continue the flow. And as it's doing so, and as I've mentioned to you many times before, one of the byproducts of photosynthesis is therefore created, which is going to be our O2 molecule. So as you oxidize the water molecule, we're going to yield oxygen as a byproduct, which is then released into the air and consumed by other aerobic respirators, such as ourselves. The other thing that I do want to mention to you also is they have this process called the non-cyclic electron flow, and basically that concentrates on the serial movement of the electrons as we start to connect the motion of photosystem 2 and photosystem 1. And when we see this happening, basically what's occurring is that we see that the electrons, courtesy of photosystem 2, will be utilized to go ahead and proceed in a sequential step within photosystem 1 itself. So basically 2 will power 1, and it turns out that by combining the actions of the photosystem 2 and 1, it will basically increase or boost the electrons to such a high energy level that it will be able to successfully produce the ATP molecules and the NADPH molecules. So that's one of the reasons why it's so important to go ahead and layer up with these particular photosystems, because you'll be able to generate your high energy electron carriers at a more successful rate. The second stage of photosynthesis is going to be our light-independent reaction. This one is going to be termed carbon fixation. Some people like to call it the Kelvin cycle. Now, I am not going to harp on all the details for photosynthesis. You might notice that this lecture isn't as detail-oriented as we did with our other steps. I'm doing this on purpose because we have so many enzymes we have to study and memorize for our electron transport chain, our Krebs cycle, our glycolysis. So we're busy. We've had a few chapters with a lot of information. So we're going to take a little bit of a detail, quote-unquote, break when it comes for photosynthesis. So basically, that's why I've narrowed down this chapter so significantly. Obviously, if you have a question about some of the details, I really do encourage you to still reach out, because I would love to talk to you about all the different byproducts and the little precursors. But for now, I definitely want you to keep in mind that photosynthesis takes place within the thylakoid membranes and the stroma. You have your light reaction, which is photosystem 2 and photosystem 1. And then the light independent reaction, the second step, is going to be termed carbon fixation. And carbon fixation, also known as Kelvin cycle, its main concern is to take the ATP and the NADPH that you were able to generate in the first stage and utilize the energy in those activated carriers to convert carbon dioxide into organic molecules that can be forming the precursor of the glucose molecule that we are after. Also some others, but glucose is the primary one that we're interested in. Along the way, it will be able to generate water as a byproduct. And the enzyme that's mostly going to be involved with this is going to be called the Robusco enzyme. The Robusco enzyme is going to help us take our carbon dioxide and start converting it into the precursors that you can see on your list right here, including the formation of your glycerate aldene 3-phosphate. You're right over here. This is one of the early precursors before we get to our glucose molecules. And in fact, what we can do as we look at all the different steps that form the cycle in a circular pattern, go ahead and take a look in the center and you're able to see what the net results are. And that's going to be that for every 3 molecules of carbon dioxide, 1 molecule of glyceraldamine 3-phosphate, say that's fast 5 times, is able to be produced. Like I said, that's going to be a precursor for our glucose molecule. And then in addition, 9 molecules of ATP and 6 molecules of NADPH are going to be consumed in the process. So we take the products of stage 1 and we plug them into stage 2 so that we can get to our food formation, which at the end of the day is going to be the ultimate goal of photosynthesis. I should also mention, and I wrote it towards the bottom of your PowerPoint, plants, if they're able to photosynthesize glucose to a level where they have access, they will go ahead and store that glucose as starch. So that is why we have food sources like, for instance, potato, that are known as being very starchy. Part of that is because they are just filled to the max with carbohydrates, lots of sugar that the plants can use during times where they might not have access to as much or any sunlight. They'll still be able to break down the stored food and generate their ATP molecules that are required for their chemical energy needs. All right, now before we end this lecture, let's go ahead and just take a look at some of the main highlights of photosynthesis, just so we're sure that we're hitting all the major points. So let me go ahead and click over. Let's see. So here we go. So it says chloroplast resembles the mitochondria but has an extra compartment, the thylakoid. That one's really important because that's the location of stage 1 of the photosynthesis. Photosynthesis will generate and then consume ATP and NADPH, check. These are the products of stage 1 or step 1, and then these are the reactants that go into the Calvin cycle. The chlorophyll molecule absorbs the energy from the sunlight. Yes, thank you very much. They'll be able to take the energy from the sunlight and start off our photosystems. Excited chlorophyll molecules will funnel the energy into a reaction center of a photosystem. We have two photosystems, photosystem 2 first and then photosystem 1. Both need to cooperate so that they convert the energy courtesy of the chlorophyll into ATP and NADPH molecules. Oxygen is generated along the way as a byproduct courtesy of photosystem 2, which has a water splitting complex or enzyme. Special pair in photosystem 1 receives its electron in photosystem 2. So that was the pairing, the coupling that we were talking about in a little bit. The second step of photosynthesis, carbon fixation, is then going to use the ATP and NADPH so it can convert carbon dioxide into a usable food source, such as the sugars, aka glucose. And then if there is excess being produced during carbon dioxide, excess sugar, the plants will be able to store it, or I should say the photosynthetic organisms will most likely be able to store it as starch, and that will be their energy-rich food that will be stored for a quote-unquote rainy day. All right, everybody, that's all I have. If you have any questions or concerns, please let me know. And I hope you guys enjoyed your unit on energy production and consumption. I know there was a lot of mention of ATP, NADH, NADPH, GTP molecules, ha 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 ha. So I'm happy that we're able to move through it, and then we'll get ready for our next exam. All right, please keep in mind I am an email away. Enjoy the rest of your day.